Hi, this program will discuss mostly manual brain extraction from a rat MRI. First of all, the scan was viewed using a program called MyPav in order to detect when the brain starts and when it ends. And then we also looked at each slice and to see where we can crop out the general brain. Using this information, we zeroed out everything around where the brain might be. Then, we imported each slice alone, and we did some processing for each slice. First of all, we used local contrast to enhance edges in the slice, and then we used this if statement. Basically, depending on the slice number, we chose the morphological processing that will be used. We mostly use a ball-shaped structuring element of radius 10 and height 10 throughout the project. However, the radius of this ball and the height changed depending on the slice. So these numbers were chosen depending on how close the brain is to surrounding objects. So for example, when the brain is very close to, for example, bulges or pieces of skull that are around it, then a larger structuring element is needed. Additionally, smoothing is a function that basically applies an averaging filter of window size 5 or 3 or any odd number, and we mostly used a window size of 5 and 3. We also did binarization. For binarization, we used either adaptive or global binarization. Adaptive binarization was only used in the first few slices because in these slices the brain was very dark. So using global binarization actually zeroed out the brain. And then, in order to select the object that represents the brain, we looked at where the brain is in most slices, and we chose a column and row number. This number also changes slightly between the if statement conditions because while the brain starts at the top, the last few slices the brain is actually in the bottom. Anyways, finally the brain was selected and then it was multiplied by the original slice in order to have the correct grayscale intensities. Now I will show you the program running.